Hello, I am Dr. Carlito Lebrilla, Distinguished Professor of Chemistry, Biochemistry and Molecular Medicine in the School of Medicine at the University of California, Davis. This lesson focuses on carbohydrates in food and their measurements via glycomics. Carbohydrates are a fascinating class of biomolecules, not only because they're a major source of energy, but also because they play many crucial and sometimes surprising roles in life. These molecules are structurally complicated, dynamic and diverse, with equally diverse functions in the overall ecosystem and in our bodies. Their functions range from communication and shaping the flavors and textures of foods, to how we digest and absorb nutrients impacting gut health. We can think about carbohydrates as the molecules that hold our world together, quite literally, with their structures and functions. We're only now beginning to understand how certain carbohydrates and food interact within ecosystems and our bodies, opening up new possibilities for designing foods and recipes with diverse outcomes for human and planetary health. Carbohydrates are the most abundant polymers in nature. Polymers are large molecules made up of smaller repeating monomers that are chemically connected together by glycosidic bonds. Carbohydrates are produced by all forms of life. It's therefore no coincidence that they are also found in food and consumed by most life forms, especially humans. Carbohydrates, along with proteins and fats, are also called macronutrients, providing us with energy. But just as you've learned that the functions of proteins and fats go far beyond simply providing energy and essential nutrition, the functions of carbohydrates are diverse and interesting. While we know much about the structures and functions of specific macronutrients regarded essential for nutrition, such as certain proteins and lipids, we know far less about carbohydrates. This has led to much confusion in diet and nutrition about the roles of carbohydrates for human health. Often, when we think about carbohydrates, we often think about solely starch. So when we feel we're eating too much carbohydrates, we really mean we're eating too much starch. Carbohydrates is a broader class of compounds that are described more commonly as starch, simple sugars, fibers, and even the dark matter of food, which refers to structures that until recently we couldn't characterize, let alone know their function. Looking at a nutrition label, carbohydrates are grouped together as total carbohydrate, total fiber, and total sugars. However, food actually has dozens of specific components. The confusion around carbohydrates stems from the lack of comprehensive tools for characterizing these compounds in terms of their structures and hence their functions. Until recently, the tools for chemical analysis provided an incomplete and imprecise structures. Only now, with advances in the field of glycomics, a subfield of Foodomics, specifically to characterize carbohydrates, do we have the capabilities to provide more comprehensive analytical characterization of these diverse biomolecule structures. You may be wondering why the study of carbohydrate is called glycomics. Glycans are bioactive carbohydrate chains. Glycanomics or glycomics, the study of all carbohydrate structure in an organism using advanced analytical technologies. The application of glycomics to food enables us to expand our knowledge in carbohydrates beyond what is on the nutrition label. For example, glycomics tools of the Periodic Table of Food Initiative, or PTFI, enable us to look at the composition of specific fibers that make up carbohydrate compositions, broken down by the type of sugar molecule and even how these sugars link together. Emerging evidence highlights that certain linkages of fibers nurture particular bacteria within the gut. As we apply glycomics to study carbohydrates, we learn that their structures are so complicated. Let's explore why. Carbohydrate chains, regardless of length, are all composed of monosaccharides. The most well-known is glucose, but there are many others we encounter in food, including galactose, fructose, mannose, fucose, and so on. There are no single monosaccharides that are found in every living being. Even now, we keep discovering new monosaccharides. Some are found only in plants, some are found only in animals. Monosaccharides can be linked to form more complex structures. Short polymers consisting of one or two monosaccharides are often called simple sugars. These include monosaccharides as well as disaccharides such as sucrose and maltose. Longer chains of monosaccharides are called oligosaccharides. Those that are very long, millions of monosaccharides, such as starch and cellulose, are called polysaccharides. To understand the structural complexity of carbohydrates, we can compare them to proteins. Proteins are made up of 20 amino acids that form chains that vary in compositions, sequence, and length. To know the protein's primary structure, we only need to know the sequence of these amino acids. Monosaccharides and carbohydrates can be linked in as many as 10 different ways, compared to the only one way for amino acids and proteins. In addition, carbohydrate structures can be branched, while proteins are generally linear. These differences make carbohydrate analysis, and particularly glycomic analysis, significantly more difficult than proteomic analysis. Slight differences in carbohydrate structures can have profound effects on their functions. Better understanding the relationship between bacteria and carbohydrates is an exciting frontier of glycomic research. For example, the study of human milk led us to the conclusion that lactose was food for the infant, but soluble fibers produced by the mother had no direct nutritional value to the infant, but was instead food for a very specific bacterium in the baby's gut. It is a specific structure of complex carbohydrate that achieves this biological mutualism. Mothers feed this one bacterium, the bacterium protects the baby. Importantly, measuring the precise composition of food was necessary toward understanding it. Now we can imagine guiding the bacteria in all of us. These two roles of carbohydrates persist in the adult diet. Starch, widely present in our foods, which includes amylose and amylopectin, is easily digested by enzymes releasing glucose, which in turn mainly is used as energy and to create tissues. The other polysaccharides, for example beta-glucan, are not digested in the stomach, but transit through the gut and large intestine, where a consortium of bacteria compete to feed on them. The carbohydrates are commonly known as fiber, and bacteria in the gut have enzymes that selectively cleave linkages in very specific polysaccharides, in turn giving other bacteria and the host beneficial compounds. There are other roles of fiber, such as blocking and activating receptor, and even blocking pathogens, and these are also an active area of research. These bacteria-carbohydrate relationships are important for plants as well. For example, in 2018, a team of researchers was studying an indigenous landrace of maize in Oaxaca, Mexico, to understand how it grows 6 meters tall in poor soil. The only clue was that it secretes a carbohydrate-rich mucilage. Why? Well, this maize grows in soil that tends to be low in nitrogen, an essential nutrient for plant growth. Over time, this plant evolved to secrete a mucilage filled with carbohydrates that selectively feed nitrogen-fixing bacteria. These bacteria take atmospheric nitrogen and transfer it to the plant. Essentially, the bacteria are helping to fertilize the maize, helping it grow in nutrient-depleted soils. Once again, it was measuring the precise structures of these complex carbohydrates that was key to discovering the functions of the mucilage. Glycomics tools had made it possible to characterize carbohydrates in deep detail, yielding the carbohydrate compositions of food, and helping us to better understand bacteria-carbohydrate interactions. These tools can determine the precise structures of the free mono, di, and oligosaccharides. They can determine the monosaccharides and linkage compositions of bulk carbohydrates and polysaccharides. More importantly, they are quantitative so that we can know precisely what parts of the carbohydrate the bacteria is eating. When we use these methods on food, we create a glycomic map of the food. We introduce a new name for carbohydrates and call them glycans. Glycans confer an bioactivity to the compound, and a glycomic analysis is the comprehensive characterization of these compounds. If we map just the monosaccharides and say 1,000 foods, it would look something like this. The blue color codes glucose. As we see, the most abundant component in common foods is starch. However, some foods do contain very little starch. This shows the linkages map of the same foods. Here we see the same colors, but different shades representing different linkages. We can monitor up to 100 different linkages. We can have a similar chart for different polysaccharides, but here the polysaccharides are better shown for a specific food. Let's look at how onions differ in their polysaccharide compositions. Here the different onions, white, yellow, and red, have different polysaccharide compositions. You can see the same in apples. The different varieties have different polysaccharide compositions. There is a rich diversity of carbohydrate compounds yet to be explored in foods that contribute to food quality, with many applications for human and planetary health. Carbohydrates are fundamental to food quality, affecting everything from texture, flavor, and shelf life to nutritional value and digestibility. For example, cellulose, a fiber found in plant cell walls, contributes to the crisp texture of fruits and vegetables, providing structure. Think of a crunch of an apple. Given their structure, carbohydrates are often used to mimic the texture of animal products and the formulation of plant-based meat and dairy alternatives. As we saw from the indigenous landrace example of maize from Oaxaca, Mexico, carbohydrates serve roles not only in nutrition, but also in ecosystems, with implications for biological resilience and health, as well as agricultural productivity. Many glycans contribute to species interactions within ecosystems, such as plant interactions with pollinators, pests, and even pathogens. For example, beta-glucans are polysaccharides that contribute to plant defense mechanism, including protection against fungal pathogens. Fructans are carbohydrates that store energy in grass and cereal crops that are important for drought tolerance, contributing to climate resilience. Cellulose, the primary structural carbohydrate in plant cell walls, can provide physical protection against pathogens and some pests, reducing the need for agrochemical input and lowering the risk of resulting residues. Not only do these carbohydrates help plants survive extreme conditions, but can also serve as targets and breeding programs to develop crops that are more resilient to climate change. In this way, Glycomics research can guide breeding and conservation efforts, helping to protect the natural processes that underpin planetary health. The application of glycomics to food systems can also help reduce food waste by improving our understanding of carbohydrate structures that affect food quality and shelf life. A longer shelf life means less food waste, which is a critical component of sustainable food systems. Further, industrial food waste, such as fruit rinds from the production of juice, can be explored for prebiotic fibers with beneficial impact for human gut microbiome. This material can be used as functional ingredient to food diets in ways that promote health without excessive resource use. As we have seen in this lesson, glycomics coupled with other fields of foodomics will significantly expand our understanding of food, revealing more functions associated with carbohydrates and how they interact within ecosystems with other biomolecules in food and in our diets, with our gut and our microbes, and with our health.